Designing Life from Scratch, a fledgling field is about to take off Part 2. The chemical components of genes and their arrangement dictates whether you spell out the makings of a lowly flea or the first violinist for the Vienna Symphony Orchestra. While the promise of gene editing has received a lot of attention of late, gene building could be a potentially more efficient technology, said Hank Greeley, director of Stanford University's Center for Law and the Biosciences. Instead of starting with the sequence of a chromosome and slowly making 100 changes in it, he said, you make a whole new chromosome with the changes you want. The chemistry for gene synthesis was perfected in 1982. But the field wasn't launched until 1999 with engineering innovations by the companies Blue Heron of Washington State and Gene Art of New York. Since then, the pace of development has accelerated rapidly. 15 years ago, a letter cost about $10, according to Robert Carlson of Seattle-based Bioeconomy Capital. At that price, a 3,000 letter-long gene would have cost more than a luxury car. Traditional gene building techniques were reminiscent of the old electronics approach to building circuits wiring together transistors one by one. But advances over the past decade in the miniaturization of fluids, automation and silicon-based technologies have increased efficiency. Just as Fairchild Semiconductor changed chip technology by layering and etching metals within silicon wafers, gene engineers are learning to build millions of DNA fragments at one time using high-density silicon chips. Costs have plummeted to 15 to 20 cents per letter. And companies such as Twist Bioscience and Boston's GEN9, vying for market share, soon will drive it down to a penny or two. This makes it possible to inexpensively string together a gene or combination of genes that are millions of letters long. And gene building that once took weeks now takes days. Furthermore, the biological code is more accurate, with far fewer typos. It's no longer Rube Goldberg type construction, said Sean Stevens of Synthetic Genomics in La Jolla, who is leading a project to alter the genes of pigs so that they can be used to grow human organs for transplant. We have locked down the commercial side with size, precision and control, and now it is time to do something with it, he said. Already, there are startling innovations. In 2010, legendary geneticist and entrepreneur J. Craig Venter synthesized a 580,076 letter genetic blueprint, then inserted it into a living cell, where it reproduced. In 2014, New York University's J.E.F. Boak built a yeast chromosome that was 273,871 letters long, then put it into a living yeast. Amiris, a synthetic biology company in Emeryville, has inserted genes into bacteria to create a potent anti-malarial drug. And the Emeryville startup Industrial Microbes plans to engineer microbes to convert natural gas into industrial chemicals. What other applications are possible? Gene synthesis facilitates how we learn and apply lessons from the natural world to improve our own, said Nancy J. Kelly, the founding executive director of the New York Genome Center. For example, she said, researchers are trying to design genes that could create new fabrics that are stronger than steel but softer than the finest wool, create renewable fuels that dramatically reduce greenhouse gas emissions, and produce meat and milk that don't require animals. It is even possible to dream of the day when doctors can save lives by replacing a deadly defect in a single gene or suite of genes. We're still 10 to 20 years away from building, from scratch, the full chromosomes of organisms more complicated than microbes, said Stanford's Greeley. Even more distant is the prospect of writing and building the entire human genome, made up of billions of letters. Meanwhile, large societal questions loom. Who will fund, control and coordinate the research and its applications? Might synthetic creatures escape into the wild, wreaking environmental havoc? Could our enemies create and spread deadly diseases? We are putting the cart before the horse. Gene synthesis still needs to be greatly vetted before anything comes to the market, said Dana Pearls of Berkeley-based Friends of the Earth. There is so much we don't know. Before we rubber stamp this as sustainable, natural and helpful, we need information and national regulations. The question is not, can we do this? But, should we do this? The field now essentially is self-regulated, with companies screening each client's mail-in order of gene synthesis for biosafety. But Congress is seeking to catch up. Introduced this session by Rep. Eddie Bernice Johnson, D. Texas, H.R. 591. The Bipartisan Engineering Biology Research and Development Act of 2015 would establish a new national program to advance and oversee synthetic biology. Just because we are able to personalize DNA at will, the Woodrow Wilson Center's Powell said, does not necessarily mean we know how to live with this power.